Strategies for disease control in organic palm fruit, apple. The major diseases affecting organic apple production are apple scab, powdery mildew, fire blight, sooty mold, sooty blotch, and the post-harvest disease bull's eye rot. All of these diseases can also affect pears. These major diseases will be explained in detail in the following sections. Apple scab is the most important fungal disease in apple cultivation and can also attack pear and mountain ash. Damage by apple scab can occur both in the field, pre-harvest, and after harvest, post-harvest, during storage. The apple scab fungus overwinters in fruiting bodies called perithecia in infected leaves on the ground. During the winter and early spring, the fungus forms fruiting bodies in the fallen leaves. Primary fungal spores, called ascospores, are released from the fruiting bodies during rainy periods in the spring. The spores are then carried by wind and rain onto leaves and fruits, where the spores germinate and penetrate the plant tissue, leading to infections. Primary infections can occur from the green tip through 1 to 2 weeks after petal fall. Symptoms appear 1 to 2 weeks after infection. Secondary fungal spores, called conidia, are produced on the lesions and cause additional infections on leaves and fruits during rainy periods. Secondary infections can continue to occur throughout the growing season. What are the symptoms and damages of apple scab? Symptoms appear on leaves and fruits. Lesions on the leaves first appear as light green circular spots that later become olive green and velvety in appearance. Lesions may remain distinct or may coalesce into larger areas of infection. If early infections are severe, the leaves may curl, become puckered and torn, and often fall prematurely. Symptoms on the fruits appear as distinct, almost circular lesions that are olive green and velvety at first, but later become darker, scabby, and sometimes cracked. Severe early infection of fruits results in deformed, cracked fruits that may drop prematurely. Late season fruit infections may develop into dark scab spots during storage. Multiple infections occur throughout the season on both leaves and fruits. The spots on the fruits lead to fruit depreciation, and the spots on the leaves to a loss of photosynthetic capacity, and over time to plant death due to depletion of the trees. Monitoring of apple scab. The fungus overwinters in infected apple leaves on the ground. In spring, ascospores, the primary inoculum, are released after it has rained. The release of these ascospores can be monitored using spore trapping. The time point of first discharged ascospores in combination with the phenological stage of the apple tree is used in forecast models to calculate the risk of infection. The RimPro model is the most widely used forecast model by producers for apple scab monitoring. To get a realistic simulation, you first need to set your biofix in local parameters. To better understand how this model works, the fundamental parts of the graph will now be explained. In the bar below the graph, Part A, the light blue bars represent the wetness of the leaves, based on factors such as rainfall in your area, leaf wetness, and relative humidity values. The dark blue bars correspond to rainfall events. The middle graph, Part B, indicates the maturing process of the fruiting bodies in the leaf litter, which is determined by temperature and humidity conditions. The brown area corresponds to spores that are not yet mature and therefore cannot yet be ejected in the next rain event. The red area corresponds to the spores that can be ejected during the next rain event. The upper graph, part C, simulates the infection process. The yellow bars, as shown on the detailed figure, correspond to the spore discharge every 30 minutes depending on the stock of mature spores, rain, temperature, and light conditions. The germinating spores on the leaves are shown as the white area on the graph. These germinating spores are still sensitive to contact fungicides. The red line is the relative number of spores that have successfully infected the leaf. The higher the red curve, the more severe the scab contamination. The orange area is the period during which the fungus has only developed its primary filament-like structures called stroma in the leaf. If you would like to learn more about this subject, please visit the RimPro website. See the reference section. How is apple scab managed? The preventative measures play a crucial role in apple scab management. When planting an orchard in organic cultivation, it is important to choose apple scab resistant or robust varieties. Furthermore, it is important to use sanitary measures to reduce the inoculum of this disease. Hygiene methods are key to reducing scab in organic production. Apple scab inoculum that would be produced from overwintered infected leaves and twigs on the orchard floor need to be removed or shredded, or their degradation needs to be promoted in autumn to prevent infection from ascospores during the following spring and summer. Pruning is also important for scab reduction in organic production. The trees should be pruned to remove infected branches and encourage good air circulation in the orchard canopy, which helps rapid drying of leaves and fruits after rain and dew. Finally, a drip irrigation as opposed to a sprinkler irrigation is recommended to reduce moisture in the foliage. Apple trees are susceptible to the disease starting from the green tip stage. It is essential to properly manage the primary infection to limit the number of treatments needed during the summer. Regarding direct control with plant protection products, preventative treatments before rainfall events and scab stop treatments after rainfall events during the early infection phases are possible. The use of forecasting models and weather stations must be combined in order to carry out treatments correctly. Preventative treatments are carried out immediately before the onset of rainfall, respectively, infection periods. Treatments should be carried out with low doses of sulfur and copper to avoid drift problems. Scab stop treatments are used to stop the disease's spread. Fungicides used in organic cultivation include wettable sulfur, lime sulfur, and potassium bicarbonate and copper. The use of fungicides in agriculture is regulated. Check which products are allowed in your country with which specifications, including use, time point, quantity. Or contact your local advisory service. It is also recommended to use copper as little as possible. Powdery mildew can affect numerous plants, including apple, its primary host, but also pear, plum, peach, and medlar. The fungus overwinters as mycelium in dormant flowers and shoot buds infected in the previous year. In spring, the infected buds break the dormancy, and the fungus infects the developing shoots and young leaf tissue. To germinate, the spores of the fungus need a high relative humidity, greater than 70%, which is commonly available in the microclimate of the lower leaf surface, at temperatures between 10 and 25 degrees Celsius. In contrast to most foliar fungal pathogens, leaf wetting is a deterrent to infection. The youngest leaves are the most susceptible, but become increasingly resistant as they mature. Secondary infections can occur throughout the season. Powdery mildew can attack leaves, flowers, and fruits. On leaves, white patches of fungal growth develop on the lower surface of the leaf. Under favorable conditions, the patches enlarge and merge to cover the entire lower surface of the leaf. Diseased leaves curl upward and become narrow, crinkled, stunted, and brittle. Infected blossoms shrivel and produce no fruit. Fruit symptoms are not usually seen unless the disease has built up to high levels on susceptible cultivars. Fruits appear with a net-like russeting. High relative humidity, overcrowding, and excessive shade promote powdery mildew outbreaks. Therefore, the trees should be planted in sunny areas with good airflow to reduce moisture in the orchard. It is also recommended to avoid susceptible varieties, remove affected shoots during the season, and affected twigs while pruning in the winter. For direct control with plant protection products, sulfur or sodium bicarbonate treatments can be performed. If problems with this disease have been encountered in previous years in susceptible varieties, pre-flowering treatments are recommended. Fire blight, caused by the bacteria Erwinia amylovora, is a severe disease of apple and pear, but can also affect quince and mountain ash and leads to tree death. The bacterium overwinters in diseased tissue, mostly at the end of branches and stem anchors. Erwinia amylovora is dispersed by splashing rain, wind, and pollinating insects, and warm and wet weather facilitates pathogen development. Trees are most susceptible to infection when they are in bloom, with susceptibility dramatically decreasing before and after the bloom period. The bacteria colonizes and penetrates the flowers, causing blossom infection. The bacteria can also directly infect young shoots through the stomata and through wounds, causing shoot infection. Noticeable blight symptoms arise approximately 1 to 3 weeks after infection, with symptoms appearing more rapidly during prolonged periods of wet and warm weather. Infections initiated in blossoms and shoots can continue to expand both up and down larger branches and limbs, causing branch death and eventually tree death. The bacteria can be further disseminated by exudates, rain, and insects and cause secondary infections, mainly wound infections. Fire blight can be observed on all above-ground tissue, including blossoms, fruits, shoots, and rootstocks. Blossom symptoms are first observed 1 to 2 weeks after petal fall. The floral receptacle, ovary, and punticles become water-soaked and dull, grayish-green in appearance. Later, these tissues shrivel and turn brown to black. During periods of high humidity, small droplets of bacterial discharge or ooze form on water-soaked and discolored tissues. Ooze droplets are initially creamy white, becoming amber tinted as they age, as shown in the picture. Infected shoot tips often curl, a symptom known as a shepherd's crook. Leaves on diseased shoots often show blackening along the midrib and veins before becoming fully necrotic. Numerous diseased shoots give a tree a burnt, blighted appearance. Bark on younger branches becomes darkened and water-soaked. Wood under the bark will show streaked, brown to black discolorations. At advanced stages, cracks will develop in the bark, and the surface will be sunken slightly. Amber-colored bacterial ooze mixed with plant sap may be present on the bark. The risk of infection should be checked using forecast models, especially during the blooming phase. The selection of less susceptible cultivars and/or rootstocks is recommended as a preventative measure. Furthermore, infected plant parts or even whole trees should be removed during the season, and vigilant sanitation should be performed in winter through the removal of expanding and overwintering cankers to remove overwintering inoculum. All cutting tools need to be disinfected carefully to avoid the spreading of the disease. The surroundings should be controlled for wild plants that could also host the pathogen. Regarding plant protection products, the yeast-like fungus Oreobastium pullulans can be used as biocontrol. This biocontrol agent should be applied prior to infection. Its effect is based on the competition for nutrients and space. When allowed, a copper treatment can be performed at bud break, when fire blight has occurred in the previous year. Copper also has an effect against apple scab. It can, however, cause some russeting on varieties. Sooty mold, also known as sooty blotch, appears on the leaves, stems, or fruits as superficial black growths of mycelium forming film. It is a disease that can affect a large range of host plants, including apples and pear trees, but also citrus fruits, olive trees, horticultural species like eggplant, sweet pepper, and zucchini, and ornamental species like oleander gardenia and jasmine. Sooty blotch is caused by several species of fungi of various types, which also change from region to region and even during the season. Sooty mold fungi can overwinter on branch tips, bark crevices, and dead wood or mummified fruits of trees and woody plants. The fungi live off honeydew, the sugary deposit forming on plant parts from the droppings of certain insects, particularly aphids and scale insects. During the growing season, the fungi produce spores that are blown and splashed to honeydew or plant exudate-coated surfaces. When conditions are wet, the spores germinate and sooty mold grows on the medium available. Warm temperatures and wet weather increase the proliferation of sooty molds. The disease usually appears in summer and proliferates until harvest, but also during storage. Sooty mold covers fruits, tree limbs, and leaves with a thin black film or crust. The dark color of sooty mold growth is due to the presence of dark-colored pigments in the mycelial threads that make up the main part of the sooty mold colonies. Many sooty mold fungi are sticky, and that helps them adhere to the surfaces on which they grow and absorb the moisture they need. The black, crusty growths can cause yellowing of foliage when the sunlight is obstructed. Sooty mold causes mostly indirect damage through the fruit depreciation because of their appearance, and the black film on the leaves impairs photosynthesis. Late varieties are more affected by sooty molds than early varieties. Therefore, using early varieties limits the damage. Furthermore, different preventative measures can be implemented to limit moisture in the orchard. These include the selection of a location with good aeration before planting, a pruning system which promotes the aeration of the canopy, a regular mowing of the grass, and the use of a drip irrigation rather than sprinklers or overhead irrigation. Direct control with plant protection products include treatments with products based on sulfur and bicarbonates. The efficacy is currently unclear as pathogens are different and vary from region to region. The black layer on the fruits caused by sooty mold fungi can be removed after harvest before storage by plunging them into hot water or brushing it off. Some special brushing machines can be implemented in the post-harvest sorting process for this. Bullseye rot, also called lenticel rot or bitter rot, is caused by fungi from the Neofabraea genera, previously called Gliosporium, including Neofabraea praeninens, alba, malicortis, and kienholzi. This disease mainly affects apples and pears. The fungi overwinters in orchards as saprophyte fungi on mummified fruits, pruned wood, dead bark tissue, and leaf litter. Spores produced on these overwintering parts are dispersed via water splashes and infect fruits from petal fall to harvest. The infection remains latent and symptoms are only expressed after 3 to 4 months of storage. Infections are more frequent in rainy years and on late cultivars that tend to receive more rain. Bull's eye rot symptoms of infection originating from lenticels show lesions that are circular, flat to slightly sunken, with a light brown to dark brown halo around a lighter brown center. The fungi usually produce lesions on the cheeks, but they may also occur around the stalk or calyx, where it enters via a wound or small crack. The brown lesions expand rapidly and numerous white pustules appear, which are the fructifications of the fungus. At an advanced stage, cream-colored spore masses may appear at the center of the lesion. It is very difficult to differentiate between bull's eye rot infection caused by different Neofabraea species on fruits. Preventative measures to manage bull's eye rot include pruning and removing cankered branches, removing rotten fruits, harvesting in dry conditions and at the right time for each chosen variety, and avoiding susceptible varieties, or shortening the period of storage. For direct control, treatments with Oreobastium pullulans or products based on acid clay or copper have partial efficacy. It is also recommended to use copper as a last resort. Post-harvest treatment with hot water is effective against bull's eye rot outbreaks. Here are the references for this presentation. Thanks for your attention.